University. We'll start in a minute or so. All right, Flora, you can start whenever you're ready.
Thank you. So, hello everybody. My name is Flora, and I've been a postdoc with Peter Andolfato at Columbia University. And today I will present some of my work on epistasis-mediated adaptive evolution. So, as you may know, evolution rates can vary even within the same protein along a protein sequence. So some region can evolve more rapidly than others. And this is mainly due to structural constraints like solvent accessibility or contact number. But it can also be due to functional constraints and selection. So for example, if some regions are under purifying selection, they will evolve less rapidly, slowly. Instead, if you have region under positive selection, they can have some accelerated evolution rate. And during my postdoc, I was interested in epistasis. And especially, I wanted to know whether epistasis can contribute to protein evolution and this variation in evolutionary rate. So what I mean when I say epistasis is that the effect of one mutation will depend on the genetic context. So, for example, in this very simple cartoon, you have the y-axis represents the fitness, and you have these initial states with two loci A and B. And the effect of mutation A on the fitness will depend on the state at the locus B. So it can be either deleterious if you have a small b, or advantageous if you have this capital B mutation. And so why do we think that epistasis can shape protein evolution? First is because we have some textbook examples where epistasis have played in protein adaptation. And this is the case for some toxin resistance adaptation. And so here I show the adaptation to epipatidin toxin. So in the poison frog, this toxin binds to the acetylcholine receptor, and poison frog can resist this mutation by mutating this site 108. But usually you don't find this mutation alone, and you have other mutations nearby that are mutated at the same time. And this is because this mutation alone can confer some deleterious effect. And similarly, you have kind of the same thing in the adaptation to cardenolides in insects. So cardenolides, again, are toxic compounds produced by some plants. And a lot of insects have evolved resistance by mutating mutations close to the at the interaction of the toxin binding. And again, so mutation at sites 111 and 122 can confer resistance, but it also implies a lot of deleterious effect that can be compensated by other mutations close by, like this 119 mutation. So in this example, we see that you have these adaptive mutations that give some adaptive fitness, but at the same time, you have those compensatory mutations nearby. And it will create this clustering pattern of mutations in a very conserved and supposed to be under purifying selection region. And so it can affect this local evolutionary rate. And another evidence or another clue for why we think that epistasis can play on evolution, protein evolution, is this genome-wide pattern. So we have shown it in a paper from the lab, but it's also been shown by other people, is that non-synonymous substitution tend to cluster. And so this is the case in a lot of different species, from yeast to primate. And in this graph, on the y-axis is the clustering score, which represents the likelihood of finding one mutations close to a first one. And on the x-axis is the distance between those pairs of mutations. And if you take two non-synonymous substitutions, so in green, you see that you are more likely to find the two substitutions close to each other within like 20 amino acids distance. And this is not a pattern that we see with synonymous mutations, which suggests that this pattern is due to selection. And we think that part of it can be due to these very local epistatic interactions. And so my main question was, are epistatic cluster widespread in protein evolution? Can we find this kind of clustering pattern in other proteins than these very specific toxin-resistant examples? And what are the consequences in terms of evolutionary paths? Because here, if I take back this very simple example, if you want to go to this initial state to a more adaptive state, you can have two options. First have this A mutation and then the B, or have the B first and then the A. And if there is a lot of epistasis, and especially negative epistasis, it will constrain your evolutionary path. So if this kind of adaptation through epistasis is widespread, can it affect these evolutionary possibilities? And so to look into that, we use a candidate approach in melanogaster. So the first thing we wanted to do is to identify those clusters of putative adaptive substitutions. So we went to melanogaster and we look at all the coding sequence in melanogaster, and we use polymorphism data in melanogaster and divergence data using Simulans, the sister species, and we create those profile for each coding sequence using this MassPRF program that give a selection estimates based on polymorphism and divergence data. So this is an estimate by site, so we can see the estimated selection per site. And so what we want to find is this kind of pattern where we have an accumulation of mutation in a very specific functional domain that has arisen in this melanogaster lineage. And this accumulation, this cluster of mutations also show a signal of positive selection. So we think that it might not be neutral, and these cluster mutations might be interacting. And so the second thing we wanted to do is to test epistasis in vivo. And so we decided to go for one candidate because it's a lot of work to do in vivo experiments. And so we decided to choose Trio because it's a rogue guanine exchange factor that is really essential for a lot of signaling pathways, and especially for neural function. And in this protein, we have in melanogaster specifically these three mutations that are present in this GHPH domain that is really conserved across evolution. And suddenly we have these three mutations in melanogaster. And this domain interacts with RAC1 and is very important for all the transduction signaling. And so what we did is that we use CRISPR to engineer all possible combinations of these three mutations into melanogaster flies. So we created those 8 engineer lines from the ancestral haplotype VNR to this melanogaster wild-type haplotype. And then we look at the phenotype. And so we chose phenotypes that are good proxy for fitness, especially viability and locomotion, because Trio is involved. Like Trio knockout are showing some locomotion defect. And so for viability, I looked at different developmental stage, and what I can see is that compared to the wild type, the ancestral haplotype is actually fine. So there is no significant differences between the ancestor and the derived melanogaster state. However, when we look at the intermediate combinations, we see a lot of defects, and especially we have 3 combinations that are homozygous lethal. So we don't have adults, we don't have pupae, it's lethal. We have one other intermediate that is quite deleterious, so it's still viable, but we think it's stable. And for the 2 other intermediates, we don't see much effect on the viability, but when we look at locomotion, so here are the homozygous viable lines that we could test. Again, we see that the ancestral is very similar to the melanogaster state, but the intermediate states are deleterious. So if we map this phenotype to these genetic networks, we see that all these intermediate states are somehow deleterious, and especially 3 of them are homozygous lethal, and 1 is probably sterile. And if we represent the same thing on this fitness landscape, what is very striking is that to go from this ancestral state to the melanogaster state, we have to go through a very deleterious intermediates. So it creates the deep fitness values in this fitness landscape. So, and especially those ones are homozygous lethal, so they can't even be like fixed haplotypes. So in summary, this by dissecting this cluster of mutations, we could see that there is a lot of epistasis, but it raises a new question for us, because we kind of were hoping that at least one path would be more likely to happen, and it looks like all those paths are quite unlikely to happen. So then we look at heterozygous individuals for the same phenotype. So we look at viability and locomotion, and we see that there was no defect at all. So there is no significant differences in viability and no significant differences in locomotion. So when we go back to this genetic network for the heterozygotes in this fitness landscape, we see that there is no more fitness value when those haplotypes are in heterozygous individuals. So one hypothetical mechanism that could have happened to go from this ancestral state to this melanogaster state, and again, it's hypothetical because the ASK state is completely fixed in melanogaster and similar in PNR, so all those combinations are not present in any species or population. But so what we think could have happened is that intermediate deleterious mutations could have segregated in the population, maybe at low frequency. And so they could be maintained long enough for like other mutations to arise. And the last mutation could have rescued the fitness of this of this final haplotype and drag this haplotype to fixation. And so for this, we do think that at least the melanogaster haplotype can have like some advantages compared to the ancestral, even if we don't see anything on the phenotype we measured. And so what are the consequences on evolution and evolutionary path? We think that dominance can kind of facilitate evolution and relax the constraint due to epistasis. And so if we go back to this example of adaptation to cardinalites, and if we go back to these 2 mutations, these 2 mutations are dominant for resistance. So if you only have one allele of these mutations, you are resistant to the toxin, but they are recessive for the deleterious effects that are associated with them. So basically, if you have an heterozygous with those mutations, it's resistant and it's completely fine. And so we think that this kind of this mutation could have been maintained in the population, give some level of resistance at the population level, and then give some time for other mutations to arise and compensate this deleterious effect, and so drag the final haplotype to fixation. And with that, I'd like to thank my lab, and Andrew initiated this work, and I'm happy to take questions if I have time. Thank you.
Thanks a lot for the lovely talk, Flora. We have time for one quick question, maybe. Right, I do.
Can you get your
See that
slides
there are
up?
no questions. Could you please get your slides up, Carsyn Bonesteel?
Yeah, sure.
So we have our next presenter, Carsyn Bonesteel from the University at Buffalo.
Hello, everyone. My name's Carsyn. I'm an undergraduate at the University of Buffalo, and today I'll be discussing my project on the evolutionary and functional impact of the human-specific ACOT1 gene duplication. So we've traditionally studied both single nucleotide variants and indels when analyzing genetic variants. These are very easy to study with short read sequencing, but they do not make up the largest percentage of bases affected when we analyze differences between two diploid human genomes. So recently, with advanced sequencing technologies, we're able to analyze structural variants such as insertions, deletions, inversions, and duplications. And while these are much fewer in count, they make up 5 times more bases affected when analyzing differences between two humans. So the gene family that I am studying produces enzymes that catalyze a reaction in fat metabolism. And this reaction breaks down activated fatty acids to free fatty acid and CoA. And among this very large gene family, we are interested in ACOT1 and ACOT2. These two genes are about 99% similar to each other on the amino acid level, but they have distinct cellular compartments. So ACOT2 has a signaling domain that transports the protein to the mitochondria, whereas ACOT1 does not have this domain and remains in the cytosol. And these genes are implicated in metabolic disorders such as pregnancy, lactation, obesity, type 2 diabetes, and non-alcoholic fatty liver disease. So my aim is to understand how these genes have evolved and what are the functional impacts on these metabolic phenotypes in the context of lifestyle changes or dietary shifts in human evolution. So this is a brief overview of the locus that I'm studying. So on chromosome 14, we have 4 genes, ACOT1, 2, 4, and 6. And previous studies have already identified that ACOT2 is a result of a segmental duplication from ACOT4. So we know that this locus is prone to segmental duplications. And specifically in humans, we have an additional segmental duplication of ACOT2, where it has been inserted upstream and differentiated into ACOT1. So to analyze the variation that we're seeing today, we have 615 long-read assemblies from HGSVC, HPRC, and the platinum pedigree. And then we've also are using about 2,500 short-read genomes from unrelated individuals in the Thousand Genomes Project. And we're using a tool called Gene2CN, which leverages unique k-mers and read depth to call the copy number of our genes of interest. So when looking at the synteny between chimps and humans, we see that the chimpanzee only has the ancestral ACOT2 gene, and it has a high identity to the human ACOT2 gene, whereas ACOT1 is lower in identical to the chimpanzee ACOT2 gene. And interestingly, we see variation in humans today in copy number for both of these genes. So from the long-read sequences, we've identified 8 different structural haplotypes at this locus. And we find 2 common haplotypes. We have the H2 is the absence of ACOT1, and this is our ancestral haplotype, as well as the derived presence of ACOT1 haplotype. And we see these 2 haplotypes at almost a 50-50 ratio across the globe. And then the 6 additional haplotypes are much less common and have arisen from secondary duplications or deletions and are much fewer in number. So when we look at high coverage archaic hominin samples, including Vintica, Chagi, and Altai, we find that the coverage level at the ACOT1 gene is much lower than the rest of this locus, signifying that the archaic hominins did not possess this ACOT1 duplication. So when we look at the haplotypes on a global scale, we see throughout these diverse populations differences in the haplotype frequencies. Pink is signifying the absence, green is the presence, and the remaining 6 rare haplotypes are clustered together as the blue. And then specifically when we hone in on Africa, we see that the 2 common haplotypes are segregating at high frequencies. And this helps us to conclude that this ACOT1 duplication occurred prior to the out-of-Africa migration, more than 45 to 50,000 years ago, but after the human Neanderthal split, as those archaic hominin samples did not have ACOT1. So we wanted to expand our data set from about 650 haplotypes to around the 2,500 individuals from 1,000 Genomes Project. And this is where we're using the gene2cn tool to call copy numbers for ACOT1. And we're seeing very similar patterns as suggested in our long-read analysis. And interestingly, we see this pattern in East Asia, where most individuals lack ACOT1 altogether. So they have that ancestral absence haplotype. And we're really wondering if there is some evolutionary force resulting in these patterns, such as balancing or positive selection. And then when we look at this more quantitatively, we see that East Asians depicted in green has a much lower copies of ACOT1 than any of the other superpopulations around the globe, including its closest neighbors, the South Asian population. So then the last thing that we've identified is we find both ACOT1 in humans and also rodents. And I'm not able to dive into all of the synteny analysis that we did to compare these, but this is a network signifying the patterns that we're able to conclude. So we find unexpectedly that human and mouse ACOTs cluster independently from each other. And this suggests that both rodents and humans have convergently evolved the ACOT1 gene. Now, interestingly, in rodents, we find a different mechanism for this. So in humans, we have ACOT2 as the ancestral gene, and after its duplication, ACOT1, the duplicated gene, has lost that signaling domain to the mitochondria. Whereas in mouse, it's kind of the opposite. So ACOT1 is the ancestral gene, and duplicated to ACOT2. And then after that duplication, ACOT1 has lost that domain. So we observe the same cellular compartmentalization, but through convergently different mechanisms, which is really interesting to us. So in summary, my work has showed that the ACOT1 duplication is human-specific. We find it absent in Neanderthals, but very high in frequency in Africa, so we can date this to prior to the out-of-Africa migration. We also find very high levels of structural variation at the ACOT locus with population-specific haplotype frequencies, and we wonder whether balancing or positive selection are shaping these patterns. And we also find independent duplications of ACOT1 in both the rodent and human lineage. So moving forward in this ongoing project, we are currently focusing on wrapping up the evolutionary history. So we're analyzing selection patterns and focusing on how ACOT1 copy number has changed over space and time by analyzing ancient DNA. We're also looking into the mechanism of duplication that has produced the variation we see today, specifically in the complex haplotypes that underwent a secondary mutational event. And then we're really interested in how this all is playing out functionally. So we're analyzing this both through computational and wet lab approaches by analyzing tissue expression data and GWAS data, as well as in cell lines with both overexpression and knockout lines to understand how the copy number variation is directly affecting function and the phenotypic effects that I discussed earlier. And with that, I'd like to acknowledge my lab and ask if you have any questions.
Thanks a lot, Carsyn. That was a nice talk. Audience, if you have any questions for Carsyn. Yeah, I think that Karen has a question.
Hitting my screen accidentally. I was just wondering if you had a favorite part of research that you're hoping to work on next. Which one of those you are most interested in?
Yeah, thanks for your question. I'm really interested in how genetic variants directly affect function. So I'm really looking forward to the next part of this project where I'll be analyzing some of the functional data that I talked about.
Awesome.
Yes, Flora, you can go.
I was wondering if you look at gene conversion between the duplicated copies, and it could help to understand the functional, the region that are functional and the one that are not, that diverge more.
Yeah, so we haven't directly analyzed that, but that is something that we're considering on how it's affecting both the genetic architecture of the locus as well as functional effects. So yeah, that's something really interesting that we're planning on looking into.
Great. Thanks again, Carsyn. So our next speaker is Ricardo Britsky, and we'll be talking on speed limitation. And however, our next speaker is the Daohan Jiang from Okinawa Institute of Science and Technology. You can start when you're ready.
Hi everyone, I'm Daohan Jiang, currently a postdoc based at OIST. Well, I also go by the English name Rex, which hopefully is easy to read. Today, I'm sharing my recent work on the principles of convergent evolution. I'm starting from this very old question. Is the outcome of evolution generally predictable? Specifically, had it been some other lineages that were lucky enough to survive the mass extinctions, what form would they evolve into? Would they evolve into something that's very similar to what we see today, given the same environments, or into something very different? Apparent in support of this view that evolution is generally predictable is a phenomenon of convergent evolution, in which similar features evolve independently in different lineages. Convergent evolution, especially that of complex morphological structures, is often implicitly interpreted as, well, resulting from adaptation to similar selective pressures. Well, actually, in many cases, we don't know for sure and can never know whether if that's really the case. But for now, let's stick with this assumption that, well, convergence resulted from response to similar selection. But even so, we can still argue or ask that maybe some of the similar phenotypes are not that similar after all, because the underlying genetic basis can still be different. For example, different genes, loci, gene families are involved in producing these phenotypes in different lineages. In that case, evolution may still be considered quite unpredictable at the more microscopic scale of biological organization. And indeed, there's considerable diversity among known cases of evolutionary convergence. We can classify them based on two metrics. Whether convergent phenotype had the same genetic basis in different lineages and whether they started from similar ancestral states. At this corner of the parameter space is parallel evolution or parallelism, where lineages started from the same ancestral condition and evolved similar molecular solutions. In this case, it's essentially like the same story being repeated. So we can say, well, seems very predictable. But if the lineages are already quite divergent when they are subject to selection, in general, they tend to evolve more dissimilar solutions, deploying different genes or loci. That is, predictability generally declines with the difference between ancestral states. Knowing that general pattern, another class of convergence would appear particularly puzzling. They are known as deep homology, where the same regulatory genes or families are recruited independently in distantly related lineages to produce functionally analogous complex structures. A textbook example for deep homology is the evolution of eyes in deeply divergent animal groups. Together, the observations include cases all over the parameter space. The question I am asking here is, when should each type of convergence be expected? When should convergent traits have shared genetic basis? So in this study, I devised a simple evolutionary model to explore some basic principles. Let's start from the genotype-phenotype map it's based on. There are regulatory genes encoding that encode trans factors. They regulate the effector genes, and the expression of the effector genes produce the phenotype. For now, let's black box this part, focusing just on the expression of the regulators and the phenotype. We can see in this study, I modeled the relationship between regulator expression level and the phenotypic trait using a linear function. We can have a function for each pair of regulator and phenotypic trait. And with more regulatory genes and more traits, the relationship can be summarized using matrices. The vector Z for phenotype, the X vector for regulator expression levels, and the C matrix for the effect of each regulator on each trait. So if the phenotype Z is under selection and the expression levels of regulators are affected by mutation and evolvable, then adaptation is essentially solving this linear system. And in principle, the solvability of the system and dimensionality of the solution space will be dependent on dimensionality of the selected phenotype and how many regulators are available. And here's a schematic illustration of two alternative scenarios. In the left one, so there's the optimal phenotype can be produced by all the expression combinations along this line. So in principle, different lineages can evolve different expression solutions to produce the same phenotype. But in the right one, so the phenotype is two-dimensional and there are only two regulators, so there's a single solution. So selection eventually pushes them all to this point. To confirm this intuition, I performed simulations where 50 regulatory genes were available and up to 50 traits can be under selection. I let multiple independent lineages evolve and then looked at the total expression variance among these lineages as a way to quantify predictability of the expression evolution. I also looked at the distance from the ancestral state to quantify like the amount of expression evolution they had. In case traits subject to selection in a given simulation happen to have high or low genetic covariance, for each level of complexity, I simulated using multiple distinct sets of traits. So I ensure I can see the effect of complexity rather than the specific set of traits. And here's the outcome. So the variance shows an interesting non-monotonic trend, increasing and then decreasing with complexity, showing a shift from parallelism away from parallelism and back to parallelism. And in this parameter space, expression variance decreased with complexity while divergence from ancestral state increased. So which means the similarity between lineages is not because the lack of evolution from the ancestral state, but because they evolved towards a common solution. Then we considered what if the lineages started from different states when selection is imposed. Here I had a like two-stage evolution scenario. In stage one, as shown by the blue arrows, they are free to go anywhere in the expression space. Then in stage two, selection is imposed. If there exists a whole line of expression combination to produce the optimal phenotype, then each lineage is likely to evolve to a solution that's close to its state at the end of stage one. But if there's only one single solution, then the selection would push them all there in any way. And again, we did this simulation, but two-staged. Like before, I looked at the total expression variance, and for amount of evolution, we focused on amount of expression evolution in stage two only. And we see a rather neat negative relationship between expression variance among lineages with complexity. So as complexity of the optimal state increased, we see a gradual shift towards deep homology. Expression evolution in stage two, on the other hand, increased. So the more complex it is, the more evolution each lineage has to undergo to adapt. In all these simulations, the C matrix or the phenotypic effect of regulators remained constant over time. So what if this part can also evolve? So if the C matrix can evolve, then different lineages have a different system to solve, and then the solution space would be different. So this would expect to have different lineages evolve more different solutions. And here's the simulation result. So the setting is exactly the same as the first one, except C matrix can evolve, and it's directly compared to the first one. So the first simulation's result here is in gray, and the new simulation is in blue. So at high complexity, allowing C to evolve generally increased expression variance among lineages, and also decreased expression divergence a little bit. And note that if we allow C to evolve, there's a greater influx of potentially adaptive mutations. So that also resulted in a better performance in adaptation. So fitness at the end are also higher if you allow C to evolve. So here's the take-home message. Essentially, conditions under which each different type of convergence is likely to evolve. So most interestingly, deep homology happens when the different lineages have like very different ancestral states, and the selected phenotype is complex enough. And this work, co-authored with Lawrence Long and Matt Pennell, is the preprint is online, and you're very welcome to check it out. And it's supported by all its internal funding to Salon unit. We also got very helpful feedback from Gunter Wagner. At the very last, I have a cute animal at the end of every presentation of mine. So in this case, this is the endemic salamander that is only found in Leucius, and we have them like right near our campus. It's really lucky to be able to see them on a regular basis. Thank you, and please let you know if you have questions.
Thank you for the great talk, Daohan. Really insightful.
Okay, I guess I spoke fairly fast.
Yeah. Yeah, we have a couple of minutes for
So what are
taking
you
any questions.
what are you looking forward to working on next?
One obvious follow-up would be to look into a broader parameter space, and in particular, what's the dynamics when the system is really not solvable? Like what kind of outcome would be? And then empirically, well, I can imagine looking into some like expression data to quantitatively see the extent of expression convergence. Well, especially like to see whether expression in novel cell types in species with similar environmental adaptations, and whether that would fit our intuition. But because I personally don't work with this kind of data for now, so that's something for more far future.
Thanks.
Yeah, thank you for the question.
Thank you, Daohan, again.